Welcome back to the Youth for Neuro podcast. My name is Pranali, and I'm a high school junior and your host for this episode. I'm joined by Ms. Sophie Rogers, a fourth-year Ph.D. candidate at the Neuroscience Graduate Group at the University of Pennsylvania and co-host of the Psych Exchange podcast. To provide some background, Ms. Rogers' educational training began with a bachelor's degree in neuroscience at the University of Chicago. She is currently pursuing her PhD at the Cortical Lab at UPenn, researching the neural circuits underlying the clinical benefits of psychedelics, and co-hosting the Psych Exchange podcast with Adrian Joe. We are so glad to have you with us today. How are you?
I'm great. How are you?
I'm also doing great. So starting off with our first question of today, I just wanted to know why exactly you chose to study your path of psychedelics and what inspired you to pursue it.
Yeah. In my background, I started doing lab internships and research projects when I was a high schooler, and I pretty much was just lucky and landed in the realm of addiction neuroscience. Also happened to like it, which is not common to like what you're doing at the age of 15 necessarily. But yeah. So I wound up just staying in addiction neuroscience labs for 7 years, high school and college. Um, And I really liked that. And I was going to continue to study addiction neuroscience for the foreseeable future. I was also a philosophy minor in college, and that questions of agency, of who has moral responsibility for things, what is the human will, how do we have a right to act that will out. I thought that drug addiction was a very special place in which to study those questions, right? What happens when the will is hijacked by a substance? So people are doing things not necessarily because they want to do those things, right? But all the same, there's some like certain level of desires that drugs can take over, um, even though you can know that you want to be doing something else. Like one of the definitions of addiction um, is when you continue to get negative consequences, right, for seeking the drug or doing the drug, but you cannot change your behavior, even though you're aware those negative consequences exist. So for me, I was doing this. I was doing like this neuroscience research program at the same time as studying all of these questions of philosophy. But in college, I had an experience with psychedelics myself, but then also these books were coming out, right? How to change your mind. And then at the same time, How to Change Your Mind by Michael Pollan for your audience. It's like the contemporary Bible, 21st century Bible, pretty much on psychedelic research and history. Um, and uh, so, of course, all these studies are now just starting to come out pretty much while I'm in college about how psychedelics are completely helping people come to terms with a terminal diagnosis, how it's helping alcoholism, um, how it's helping other forms of drug addiction, depression, PTSD. And prohibition was just starting to end, like the cracks were in the walls. Um, at that same time, once again, I started studying a core neuroscience class at the University of Chicago called Sensation and Perception, which is all about how the brain constructs perceptions from like very basic data that it's getting from sensory neurons. So, if you think about light, you're getting a binary signal. Is there a photon or is there not? How are we turning this like matrix of binary signals into the beautiful, rich 3D experience that we're having? So I was learning that at the same time as I was studying phenomenology and philosophy, which is a completely different way of trying to answer the same questions. What is experience? How is experience constructed? But from a different axiom. In neuroscience, you come from the perspective that the material world pre-exists everything else. So the data that you're getting is like real. And then phenomenology comes from this thesis of idealism, where basically actually matter is not something you can take for granted. It's not even necessarily fundamental. It may not even be there. It may just be this an aspect of how of the structure of experience. And so I was taking these two classes that were fundamentally pitted against each other in the most fundamental levels. but we're coming to some similar conclusions about the structure of our experience. And then at the same time, I had my experience with psychedelics. All of this was kind of in a crucible together. And of course, psychedelics disrupt your ability to create experiences, to synthesize your sensory experiences in the same way. And then also massively alter the kinds of ideas that you can bring into that you can hold in your mind and do allow you to change your behavior rapidly. When I got to Penn, it was the pandemic, it was 2020. Not a lot was going on. I could barely go into but as a result of this, I was able to just get a little bit of funding to do a virtual seminar series with the Mind Core Institute called New and Noteworthy in Psychedelic Science. And so my PhD advisor, at the time, I was planning to take Bible study opioids. He was like, wait, you're interested in psychedelics? I was like, you're interested in studying psychedelics? And it was kind of like the spider, and it just sort of worked out. Yeah, so I got really, really lucky. Now there's a lot more people with licenses to study psychedelics, but back then I really thought it was going to be like 10 years before I would be able to investigate that. But it was just a mixture of a bunch of different influences, for sure.
Okay, yeah, thank you so much for that. And my next question is, a lot of your research, as you said, also it's about psychedelics and also neural circuits. And so, how do those neural circuits, like, how exactly do you collect data on neural circuits, and how do you make sense of that data as well?
Oh, well, if we could answer the second question easily, that would be amazing. But no, I can absolutely tell you.
Yeah.
So there's many ways of looking at neural circuits. So if you think about, what might someone say is a fundamental unit of a neuron, or microglia, astrocytes, but from the canonical view, the neuron, right? All these neurons make thousands of connections to each other. And some of these connections have many synapses, which are the connections between neurons. Some have very few synapses, so they're different weighted connections, right? With each of these neurons. Each of these neurons expresses different sets of mRNA and therefore different sets of proteins. They are sensitive to different types of stimuli, to different types of neurotransmitters. And so we can break down these cells, in theory, we can break down these cells into something called cell types. So let's say you have a dopaminergic cell. These are the cells that express and transmit dopamine, right? In theory, you could have a cell expressing something that it doesn't also transmit. Typically for dopaminergic cells, you could classify cells in that way. So you can identify cells based on where they originate, based on what neurotransmitters or what receptors they express, or what stimuli they're sensitive to, and then where they project to. And the reason I was really interested in this is because recently there's been a lot of technologies that have developed. Well, the reason we're interested in this is because, this seems to be how the brain is built, so we want to understand that. However, the reason this could be interesting in terms of the clinic and humans one day is we have now a bunch of tools where we can target specific populations of neurons based on their cell type. So if a neuron is expressing their gene for the dopamine 1 receptor, for instance, right? You could inject a mouse in the striatum with a virus that tells the cells to express a genetically engineered construct under the dopamine 1 receptor promoter, which would mean only cells that are already expressing that receptor would express that construct. And what these constructs are things like optogenetics and DREADs. And DREADs are designer receptors exclusively activated by designer drugs. And basically, this allows us to have these cells express receptors that would never normally be endogenously activated in the body. So you would make these cells express silent receptors. What this means is, though, using a certain tool, so it's optogenetics, we put a laser in the brain, and these cells become light responsive. With DREADs or chemogenetics, we can administer a drug that is inert throughout the rest of the body, but would activate our designer receptor. And we can tell those neurons at will to turn themselves on or off, right? And this allows us to really study the necessity and sufficiency of different functions. in different brain areas. And this will hopefully one day allow us to perform precision psychiatry in humans. Or let's say if we find out that psychedelics exert their really pro-therapeutic effects through this pathway. That would be amazing if we found that, but through this pathway. Maybe we could then engineer a drug that only targets that pathway if we identify that cell type, right? And that way, people who are schizophrenic or have bipolar mania don't need to have the hallucinogenic effects. Maybe that way we can just make sure that no one ever has a panic attack while they're on psychedelics. We could maybe titrate the levels of suggestibility, right? We can make it safer for people to take by themselves. So there's a lot of reasons people are interested in circuits. However, my work focuses on monitoring activity levels. And you can do this with special types of electrodes, but I typically do it with something called single cell calcium imaging, or in vivo imaging. And basically what I do is I inject one of these viruses I was telling you about. I inject one of those viruses that expresses GCAMP8, which is a genetically encoded calcium indicator, or GECI. Lots of acronyms here. But it makes these cells glow whenever calcium. And this is a really useful property because basically calcium only flows into neurons when they're actively undergoing action potentials. Otherwise, there's very low calcium in the cytosol of those cells. So now we have a way of making these neurons glow whenever calcium flows. So then I just implant something called a miniscope, or micro endoscope. And that's basically a microscope / camera that a mouse can wear on its head, and it can freely behave, it can run around and do all the behaviors that you're trying to monitor. But I can actually create videos of these neurons firing during behavioral tasks and while they're on psilocybin. So that's the major tool that I've been using to look at circuits.
Okay. And also pivoting here, what would you say are some of the disadvantages that come with the clinical and even just normal use of such psychedelics?
Yeah. Okay. So I think there's definitely many disadvantages right now. So I think the main thing is because of our I do think there are probably solutions, but right now there's rules that you have to have 2 therapists in a room with you the entire time you are undergoing psychedelic assisted therapy. Because you're undergoing a very hallucinogenic experience, a highly emotional experience, it's probably not wise, given that this is being administered in a medical context, to allow a person to ever go unsupervised while they're on psychedelics for a medical purpose, right? This means though that it's incredibly expensive. If we know that just going to therapy once a week for an hour is expensive. For psilocybin, this is probably 8 to 10 hours total of having 2 therapists employed. And it's very expensive up front. The thing is, it would cut costs eventually for insurance companies to just do this rather than paying for your SSRIs every month. But yeah, I think that's a huge disadvantage in terms of actually rolling it out, which is why you have people like David Olson's lab at UC Davis looking at how to make them adjust psychedelics so that they're not hallucinogenic. However, I do think that there's something lost in the experience if we've totally just cut that out. So I think finding some other creative solutions would be nice. On the other hand, the other disadvantages are the people that have comorbidities. So when you have multiple disorders at once, which especially for mental health is extremely common, it's rare that maybe anecdotally, but I think it's rare you find a case of anxiety without also depression or vice versa, right? However, if people also have bipolar mania, schizophrenia, or psychotic-like disorders, the hallucinogenic experiences and likely some of the serotonergic pharmacology can induce a long-lasting psychotic episode and may even induce psychosis in people who have predispositions. So these are things to keep in mind, uh, especially like if people are going to use it recreationally, know your family history, and don't do it unless you feel that it's right for you, because it's not supposed to just be fun. It's rarely just.
Okay, and this is just a question that I had from reading about psychedelics. And so, how more specifically, how are serotonergic psychedelics different than SSRIs? Because they both have serotonin, and I just don't exactly know how each is different from the other.
Yeah, no, that's a great and super important question. And there's aspects of that question people are actively working. So first, so the major difference is that serotonergic psychedelics, such as psilocybin and LSD and DMT, they all bind to the serotonin 2 receptors directly. So they're like a surrogate for serotonin, right? SSRIs don't work that way. SSRIs block the reuptake of serotonin by inhibiting the enzymes that do that, right? By inhibiting the uptake of serotonin, you cause a buildup of serotonin in the synapse, and that's what's stimulating the receptor. So you don't have all the pharmacology that's going on in terms of the serotonin receptor interaction is the same as it should be in dogs, right? But psilocybin and LSD are kind of taking serotonin's spot in the brain when you take those. And they have different chemical structures that cause different sequelae inside the it's bound uh, to that receptor. So receptors, everything is based on proteins, and they all have these physical structures that are really important. Physical and chemical structures really important to understand what they do. Serotonin receptors are called G protein coupled receptors. So this is the receptor. So I'm holding my hand in like a C shape, and I have my other hand, which is which is LSD. Let's say this is LSD. And I'm holding my hand in a ball. When LSD binds the serotonin receptor, that receptor grips around it, but LSD has this part of it that locks in this flat of the serotonin receptor, and it literally can't get out of that receptor for a long time, which is why LSD lasts so much longer than psilocybin. It literally keeps itself locked into the receptor, and it causes different chemical reactions to happen inside the cell, or rather different proportions of those reactions than serotonin does. And we think this could be some of the reason for its effects. Importantly, though, LSD, psilocybin, and all of the psychedelics are what we call promiscuous drugs. So they bind to many, many receptors. They just happen to bind to the serotonin 2 receptors much more. However, there is a great mystery as to why SSRIs they don't work as well, because they also cause BDNF release, which is a neurotrophic factor in the brain that makes neurons grow new connections. They stimulate BDNF release. They cause dendritic branching and dendritic growth, just like psychedelics do. But the behavioral benefits don't come immediately when that happens. And we do not know why that is. This is still a very active question.
Okay, got it. And so would you say then that those serotonergic psychedelics are more specific in nature, that they in inducing those like pathways, is it similar to the way serotonin acts, like how, let's say, LSD binds to a receptor? Is it the same type of chemical reaction that would occur if serotonin also binds to a receptor?
So yes and no. So, but okay. First of all, if anyone is really interested in these questions, they should check out work by David Olson's lab, by Brian Roth's lab, by the Nichols labs. Um, I'm not the expert in the actual chemical processes that are happening, but I can tell you what I do know. But basically, the serotonin 2A receptor is bound to a few other proteins inside the cell, right? When serotonin binds it, that receptor basically tells those proteins to go off and do your other things, right? So it like dissociates from those proteins and it starts to cause a chain reaction, which is what leads the cell to become more or less transcriptionally active, fire more, whatever the downstream effect. So there's a set of chemical reactions that can happen when LSD and psilocybin bind that receptor, as well as other agonists of that receptor, it causes those reactions to happen in different proportions. So, for instance, the G protein coupled receptor has a G protein that's that is attached to it on the inside of the cell, and it also has something called beta arrestin that is attached to it. And there's a whole line of research going on right now about the ratio of G protein to beta arrestin activation in response to psychedelics, and as well as non-psychedelic serotonin agonists like glyceride. And we are actually starting to think that the ratio of that activity might be really important for the hallucinogenic aspect of serotonin. So there's more work going into creating biased ligands that might induce the neuroplasticity without the hallucination.
Okay, that makes a little bit more sense.
Yeah.
And another question I had was, how exactly do psychedelics impact the adverse pain effects? So I mean, I don't need the exact pathway, but how exactly does it work is what I'm wondering.
I mean, look, that we just don't know. Honestly, the more
Okay.
that I'm seeing in mice, the more mysterious. So basically, so I actually work in a pain lab, but I don't study pain. But now I do know a lot about it because of my colleagues. So there are three aspects to pain that are really that you can study separately in the brain. So one is the sensory aspect. So if you cut yourself while you're in the kitchen, you have the sensation of the knife cutting you. So that's like nociception itself, the basic sensation. Then you have the affective access. It's affective, so that has to do with your emotions. So it's the emotional response. Like when you're in pain, you get sad, you get angry, you get scared. You have all of these things like, oh, just ruined my whole day because I stubbed my toe, right? So that's the affective side. And then you have the cognitive side. You start to ruminate about your injury while you're healing, right? You start to think about how you need to reorganize your life and taking care of yourself while you're on the injury. So all of these things happen in various parts of the brain, right? Sometimes they all happen at the same time in the same place. Sometimes they happen separately. I suspect, I suspect that psilocybin does not so much affect nociception, or psychedelics don't affect nociception. They don't make you pain blind, which would be very problematic. I do think we would know that by now if that happened. I suspect that it changes people's relationships to pain. So, for instance, if you have a chronic back injury, it's one of those cases where you no longer have a back injury. You just have this like phantom pain. It could very much help with that like pathological, affective, and cognitive aspect. However, this I don't think is right in all scenarios because as we know, psilocybin has been effective in like cluster headache studies. So I wonder, so psilocybin also activates these inflammatory pathways. So I wonder whether it could be a whole like non-serotonergic aspect of psilocybin if it really is helping the pain perception itself. That could very well be its anti-inflammatory effects. But
Okay.
yeah, mystery. Please come work in the field. We need you.
Yes, I would love to. But another question is, you're the first scientist in the 21st century at Penn to study the effects of classical psychedelics and also serotonergic psychedelics. And so you also created, you also you sorry, you did this event called the MINS Year of Psychedelics, where you organized and supported along with MindCore. And so what was the biggest scientific breakthrough you learned, and what exactly would you take away from that event?
Um, yeah, so that event, so MINS stands for the Mahoney Institute of Neuroscience. I forget what that is if I'm being totally honest. But yeah, that's the institute at Penn that organizes a lot of neuroscience events and seminars and gets pretty much everyone in our department together. And every year they have some theme in neuroscience. So I didn't help organize this, but I was very happy about it. I had organized the Mind Course seminar series a couple of years before. But basically, every year they choose a theme and they invite big build speakers to come. yeah, which is, I mean, it's an incredible opportunity for me because most of the prominent psychedelic researchers in the US, not all by any means, but many have been located in California. They're at Johns Hopkins, you know. So it's was really nice to be in the same place as some of these speakers. So David Wilson came, Fred Barrett came, Robin Carhart Harris, who is now, I believe, at UCSF, but was at Imperial College London. Um, so yeah, it was a fantastic opportunity. So I got to present work that I started where it was a year ago, which now I'm like, I'm not showing that to anyone else now because I actually figured out the things up this way. But it was a great opportunity. About 100 people probably came at least, and my PI, my advisor, gave a talk on my work. And yeah, it was great. I think the coolest thing that I learned there was David Olson's lab is working on creating agonists for ser that are sorry, not just how I was talking about biased agonists for the serotonin 2A receptor earlier. Well, psychedelics also bind with high affinity to the serotonin 2C receptor. And the 2C receptor, like its role in this is not, I'm like trying to chip away at that question right now. But basically, what we know about serotonin 2C receptors is that they're on dopaminergic and they seem to gate dopamine activity. So we're wondering if the serotonin 2C aspect might be the reason that psychedelics aren't very addictive, right? It's strange because they can cause these states of bliss and euphoria. And in every other drug, that usually means it's going to be a little addictive, right? But we really don't see any evidence of that with serotonergic psychedelics. So basically, he's trying to design agonists that are also equally have equal affinity for the serotonin 2C receptor, because we're thinking this might have important anti-addictive and perhaps antipsychotic properties as well. So a lot of that precision psychiatry stuff that I was talking about before. So seeing that was probably my favorite part of it. Yeah.
That's super cool. And also from that event, and even generally from your own experiences, how has studying psychedelics influenced your own personal perspectives on neuroscience and also psychedelics in general?
Hmm, that's a good question. I don't know that studying psychedelics in particular is anything that changed my perspective. I mean, considering your audience right now, I think if I'm being honest, what has really changed my perspective is uh, because of the type of data I'm collecting. I'm collecting data from thousands of neurons at a 20 hertz frequency for like half an hour over 5 days. I mean, it's a massive amount of data that I'm looking at. And I naively thought that I would be watching these neurons, I would play my fear conditioned time, and the neurons would turn on, and then they'd go off, and everything would be good, and it would be easy to track. No, I was so wrong. I couldn't have been more wrong. And it wound up that I had to teach myself data science in MATLAB over the course of about a year. And I was really lucky. I wound up going to a summer school. So they have all these summer schools for grad students, which are just awesome neurocamps. I actually had the time of my life. It's just you're spending 12 hours a day with this group of people that are in an equal like level as you. They're all PhD students or postdocs, but they're all also ignorant of this one thing they need to become better at. So I mean, it's a fantastic opportunity. But yeah, so I wound up having to teach myself how to code. Like I had to teach myself data science, and I never saw myself as a math person ever. So I was extremely intimidated by doing all of that. But I have to say, the ability to teach myself something through trial and error in private, so I never had to worry about getting it wrong because I could always try again. Um, and it was just a wild experience. And for me, branching out into that new field has very much changed my perspective on potentially where my career could go, and what I'm even capable of. So, yeah, I mean, for me, that was probably the most transformative part of this experience.
Okay, and this leads me actually to my last question of today, which is what would be your most important actionable advice for a high school student or an undergrad listening to this podcast right now?
Yeah, okay. My advice would be first of all, basic, but A, cold email everyone whose lab you want to work in. Do not worry about whether they want to hear from you. Don't just write an email saying who you are, why you like them, and how you could be useful to them. Just send it and follow up once a week.
Okay.
Because worst case scenario, if you're bothering them, they will never remember that. They will never remember who you are if they don't respond to you. But they might finally respond to you. And then choose a lab that has the technique that you really love before you choose a lab that is the topic you really love. I think that's probably if I had to change anything about what I had done when I was younger, I just felt like that topic seems shiny cool and that topic seems shiny and cool. But then I wound up like not gaining really genuinely new skills until grad school. And I had 7 years to do that. So I think that, yeah, I think that
Yeah.
that's the advice.
Very good to know because I myself have also been cold emailing people, and I haven't really been getting a lot of luck with getting responses and stuff. And so I'll definitely keep following up with them and then
Yes,
see if something comes.
PIs, PIs are so busy.
Mhm.
And so it just, you can't like, they also, for the record, there's actually a very important grant that's coming up today. So PIs are going to start responding more. So just inside.
Okay.
Yeah.
Okay, good to know. But yeah, this actually concludes our podcast for today. So thank you so much for joining us today, Miss Rogers, and make sure to follow her Instagram account at miss.sophie.ann and check out her podcast at psychexchangepodcast.com. If you enjoyed this podcast episode, be sure to follow us on Youth for Neuro on Instagram, Spotify, and LinkedIn, and check out our website linked in our bio. Until next time, this is Pranali signing off.
Very
Okay.
quickly,